This video is going to explain the directions for the lab on vision. So first off, let's look at hey guys, I'm making a video. First of all, there are a couple different documents posted. The first is the scanned version of the lab. So you're going to go through it. As always, the first part has a lot of information in it. And then there is a second document that you're going to put your answers in. So these two documents go together. So you're going to read, you're going to look at the different parts of the eye. If you have somebody at home, look at the side of their eye like this so you can really see the cornea structure and the pupil and the iris and the sclera and how they're arranged together. And then you're going to identify the accessory structures of the eye. P.S. They're up here. And then you are going to you're going to say, ask a student to look to the left. The student, you're going to need a friend at home to do this, a parent, sibling. And so you're going to ask them to do that. You're going to ask them to look to the left. And then you are going to use this chart to say what muscles they are using for that. And then you go to this sheet, and this is activity 1, so then you write your answers in here and here. When I post this on Google Classroom, you will just be able to type directly into that. All right, activity 2. Activity 2 is looking at the retina, so we're all going to look at the retina together. This is a H&E stain histology of the retina. The light is coming from the top to the bottom. So as you look at it, this is the sclera, the white part of the eye. This is the choroid, the part that has the blood vessels. This is the pigmented epithelium here. This is the outer segments of the rods and cones. These are the parts that depolarize when they get enough light. This is the nuclei of the rods and cones. This is the nuclei of the bipolar cells, which are between the rods and cones and the ganglion cells. So these are the ganglion cells, and there are some nuclei, but then the rest of them are axons. And if you look, nope, not that. Those axons, so hold on. Those axons here go out and they exit. These are them where the mouse is. And they exit out the optic nerve, which is also here, is the optic disc. And the optic disc is the place where the optic nerve exits the eye, and that's the blind spot. We will talk about finding your blind spot in a minute. All right, so that's activity 2. Activity 3, you are going to go and watch an eyeball dissection. There is one of a cow eyeball here. And there's a playlist of human eyeball dissections. The human eye dissection is very graphic. It starts with an eye and a face. So there's a trigger warning on that. So that's for the very brave or interested. There's no shame in just doing the cow eyeball dissection. That is completely cool. If you do the human eye dissection, the questions are about the cow eye dissection in the lab. So go back. There's like 3-minute versions of it on YouTube. So then you're going to answer the one question for that is, what is the point of attachment in the retina called? P.S. I also made all the questions that you're answering in the body of the lab in bold, so make sure that you're answering all the things in bold. So that's what you're going to do for that. Activity 4. Oh, not this. Let's get rid of that. Activity 4. So activity 3 is the eye dissection. Activity 4 is the visual pathways of the brain. The questions are on page 370, but this is the diagram you're looking at. So essentially what you're saying is this part here is your field of view. And it's taken in by these two eyeballs. Please don't suck your popsicle right here. Oh my goodness. Sorry. So here's your field of view. And the vision comes in. If you have full vision, you are seeing this with both of your eyes. And then it talks about having a lesion. So say if you had a lesion here in your left eye in the optic nerve, it would block all of this vision. And so if you go back and see, what would you miss? You would miss the peripheral vision of your left eye. You would also lose this binocular vision from both eyes, but you would not have vision in the left eye. So what you see here is this part of the, so it's sort of the far right part, so sorry, the left part of the left eye, because you're looking down on it, it's the patient's eye, is going to go, that at the optic chiasma stays on the left. Excuse me, Miriam, I'm making a video and I need you to be quiet. And then on the right hand side, there is that from the most medial part of the eye, that part crosses over at the optic chiasm and goes and is viewed on the right hand side of the eye, and vice versa. So it talks about if you have a lesion here in the optic chiasma, what happens, what would happen, and you're just going to trace that back. If you lose vision on one side, just trace it back to what you're looking at, where those fibers are. Do you lose periphery? Do you use it on the left? Do you lose it on the right? So you're going to use that as a indicator. Psst. Shh. Okay. That's activity 4. And then you answer the questions here. Activity 5. You are going to go to this website, and we're going to do it together, about testing your blind spot. I like this yellow-green one, and I'm going to talk you through this because this is not particularly easy. So the blind spot, you have this on your screen, and you are going to close your left eye. You're going to eventually have somebody, once you figure out how to do this, you're going to have somebody measure the distance between your eye and the screen. That's the data you're collecting. But just to find it, you're going to close your left eye, and then you're just going to move your face. Oh, sorry. You're going to close your left eye. You are going to focus your vision on the green plus, the plus in the green box. But then you're going to move your face towards the plus, and you're going to pay attention to the dot in the yellow. So it's in your peripheral vision, so you're still focusing on the green plus, but you're paying attention to the yellow dot or the dot in the yellow box. And as you get there, mine is about, I don't know, 10 inches away, the yellow dot will disappear. So again, you close your left eye, you're with your right eye, you focus on the green plus, but you're paying attention with your peripheral vision, but with your mind, you pay attention to the yellow dot, the dot in the yellow box, and eventually it goes away as you move your face towards your screen. And then you're going to repeat it on the other side. On this side, you're going to close your right eye, you're going to put focus on the dot in the yellow box, but that's where the focus of your eye is, but your attention on the plus in the green box, move your face towards your screen, and Bob's your uncle, the green plus will go away over time. All right, so that's how you do that. Measure the distance between your eye and the screen. That's easier done by your partner, whoever that is, and that's the data you're collecting. Activity 6, near point accommodation. You're going to do this in your lab. You can use the tip of a pen or something. You're just looking at something very small, and you're going to measure with your left eye and your right eye where your near point accommodation is. If you would like to test someone else for this lab, you can do that. That's fine. Oops, let's make that whichever. All right, there are 2 choices for activity 7. One is this way of measuring your visual acuity. This one, you think you have to, you have to accept the things and do it. A meter away is about as far away as you can get without and still touch your mouse. You cover one eye. This is telling you what eye. I think it's the right eye. And as you go, the E's get smaller, and you point which way they go. And if you can't see it, you say you don't know. Or you guess. And then you continue with your right eye covered. Oh, that was supposed to be the left eye. And you keep doing it. This one's good because if you keep going, then you can do the astigmatism testing, which is the next activity. So when you get here, do the astigmatism test. If you want to be an ophthalmologist someday or an optometrist, and you have a printer, I suggest you go to the Snellen test and print out a Snellen test and you put it in your house. You know what? Everybody needs more fun in their lives. Follow the directions for printing this out. Stick it on a wall someplace, and test and see if you can see what everybody in your house's vision is. If you wear contacts or glasses, you should do this both with and without. So, don't take your contacts out just for this occasion. Wait at bedtime, take your contacts out and then do that testing. So you're going to do that with both eyes, one eye covered for each of them. Activity 8, you're going to continue from here. So if you did, no, not that one. If you already did this one, you are going to continue and start the test for astigmatism. You cover one eye. You see if some of these look bolder. I will say that for mine, I've never had an astigmatism before, but today I tested positive for that, so that's fun. Better astigmatism than COVID, right? So you're going to do that on that same website. If you didn't use it before, it's linked here. Activity 9, you're going to test yourself for color blindness. You go to the website. At the end of this, they try to make you put your email in. But then below where you provide that, there's just like, no thanks, I just want my results. Just click on it and it will give you your results. So there are a bunch of different kinds of color blindness, and they will tell you which one you have, if any. Activity 10, you're going to have to scrounge around your house for a narrow container. We usually try to dunk a pencil in a test tube that's in a test tube rack at school. So if you can find something small, the thing I could think of is the structure in your bathroom that you use to put toothbrushes in. It's usually like a ceramic thing that's mounted to the wall. If you have one of those, that would work perfectly. Try to dunk a pencil in there with one eye closed and really kind of wait for your vision to accommodate and try to use that. So whatever you use, put it in the space here and then describe how the difference with and without binocular vision. Activity 11, you need a flashlight and a person who is in your household because you're going to check their photopupillary reflex, follow the directions. Don't shine the light in their eyes too long. It can damage your retina, so don't do that. And then you're going to do an accommodation exercise. If you did this with brain and body, or at least if you did it with brain and body in my class, you did it with reading, and they actually really find your accommodation point. But go ahead and do that. And then the convergence reflex, you're going to go ahead and follow the directions in your lab. As we scroll through, we are not going to do the ophthalmoscopic exam. I have an ophthalmoscope, and even I am not going to shine this in my children's or my husband's eye. Spending too much time looking. See, do not examine the macula for longer than 1 second at a time. That's because that can cause visual damage, and that's not good. We don't want vision damage. All right, so that brings us to the written part of the lab. I did not type this all out because, you know what? I got things going on, and so I didn't do it. But I did give you answer places, and I tried to sort of summarize what it is that we're talking about. So you just type your answers into the spots. So particularly for the matching, I didn't write out the question or anything. You do that. But I did type out some of them, so it's, I feel like it did fully half the work for you. All right, so you're going to do all this. You're going to submit the whole thing by Friday. You need to actually submit it to me by Friday, and then you're going to make a copy of it, and I'll talk to you about doing that in class. And Friday at noon, we are going to correct it together like the good old days. And I'm just going to keep saying good old days because it makes me feel like it. All right, that's the end of this video. If you have other questions, send me an email and I will get back to you.